Okay, thank you very much. It's a great pleasure to have Martin from the John Innes Center, and he's going to talk about integrating analog and digital models of gene expression. Over to you.
Well, thanks, Peter. Well, thanks for inviting me. Very nice to be here. Such a distinguished audience. Thanks for coming. So I'm going to be talking about analog and digital modes of gene expression, which I think will be a type that's new for everyone except Sarah, who heard a probably incomprehensibly shortened version of this talk in Cold Spring Harbor, where everyone only had 15 minutes. So, as you can imagine, all the talks got compressed to oblivion, more or less, actually. So, right, and this will be about... Thank you. But so this talk will be about sort of self-fates in a manner of speaking. So hopefully that will become clear by the end. Okay, so first of all, I need to introduce some background. Now, this I think will be clear for some of the people who were here last week, the condensate week, when epigenetics was part of that. But for those of you who weren't here, I think it's lots of you, maybe those of you who are new to this, a lot of this talk is going to be about epigenetic inheritance. So what is that? Oops. So epigenetic inheritance is inheritance of phenotype from one generation to the next that doesn't depend on the underlying DNA sequence. So it's information that's not held genetically in the DNA sequence. And it comes in two varieties. There's a strong variety, which is transgenerational, and it's really through the germline. And there's a weaker form of epigenetic inheritance, which is not through the germline, but it's just through cell division and mitosis, such that a given gene expression program can be remembered and then passed on to the progeny of a given cell. So that a certain cell type can keep manufacturing cells that are of the same cell type. And I will be talking about the second type in this talk. And as I say, it's vital for differentiation, and it's also vital for environmental response. And this talk's actually going to be mostly about epigenetics in a context of environmental response. So what are the questions that my group's interested in? I'm interested in how epigenetic memory states are switched and maintained. And my argument, I would argue that despite many decades of intensive work, we really don't understand this very well still in biology. How do you set up these memory states? How do you maintain them? How do you switch them appropriately? There's some very deep issues. We understand a lot of the biology. We know a lot of the component parts of these systems, but I don't think we really understand very well how they all fit together to form, this wonderful system that's able to maintain these expression states despite many perturbations and over very long periods of time. So what our approach is going to be, and I've been trying to do this for quite a long time now, embarrassingly long perhaps, is to integrate theoretical and experimental analysis. But what's unusual about this is I'm going to be focusing mostly on a very small number of targets, really just one target for most of the talk, although we will go genome-wide briefly at one point. Whereas most of the field is just genome-wide all the time, really all the time. And that means that it's very difficult to get away from correlations, and start to get to the causation of what's really going on here when all you're ever doing is measuring everything at once and correlating things. Okay, so we're going to try to write down some simple but hopefully realistic models. And I guess over the years that I've been doing this, my definition of what simple is slowly becoming more elastic. They're not quite as simple. I am still a theoretical physicist, but I've had to kind of fight with myself to allow more complexity to be in the models than I probably would have tolerated 15 years ago. Right. Okay, so this is going to look just embarrassing, right? It's almost the simplest thing you can possibly write down in gene expression. And how do you quantitatively control gene expression? And here you've got some input signal, and later on we're going to see that's an environmental signal, so it's going to be cold actually. And you get some output in terms of gene expression. And what you want to understand is, well, how do you turn that input signal into the gene expression output? And you're probably thinking at this point, well, you can't write down anything that's more simple than that, right? It's just a linear relationship between input signal and output signal. So what on earth am I going to be talking about? So, but the point is that there are two ways of doing this, which I want to highlight, and then ask, that's the analog and digital in the title, and then ask how these two modes of regulation actually interface with one another. That's really what the talk's going to be about. Because if you want this, there are two ways of doing it, although of course you can interpolate a bit between them. And that is the analog and digital way of doing things, right? Digital meaning, on-off switch on a light, so it's an all or nothing response. Analog regulation, think dimmer on a light. You can turn it up and you can control the intensity of your light in a continuously varying fashion. So if you apply this to gene expression, the analog one, first of all, what you imagine now is you've got some gene that's been regulated, perhaps it's under inducible control by an experiment, perhaps it's just been natively regulated, but as the input signal rises, you just increase the amount of transcription at every single copy in the genome roughly similarly. And the system can go from a very low expression to a high expression, and you basically just change the transcriptional output at each locus in a continuously varying way, as illustrated here. So that's smoothly varying. And if you want to have 50% expression, you just have all your gene copies on at roughly half their maximum output. Right, very, very simple. But of course, there's the other way of doing it, which is the digital regulation system. And here, although you'll get the same output at a population level, at an individual locus, of course, it's very different. Because what you're doing now is you're switching individual loci on, or they're sometimes off and sometimes on, and it's the fraction that are on that is being changed. So as you ramp up your input signal, each individual locus is either completely off or completely on. It's simply the fraction which is changing, right? Okay, so that's all or nothing. And if you want 50% expression at the population level, you turn on half your copies, and the other 50% are off, right? I think to this audience, I hope this is quite intuitive, right? You know, and you're familiar with some of this. You wouldn't believe how hard it is to get those two concepts over to a biological audience in epigenetics, right? Just apart from Sarah and Laura, you could get a free pass. Okay, Jason, and other biologists in the audience. I mean, this is my experience in Cold Spring Harbor and other places about how hard it is for them to get this idea, actually, about this, not so much the analog one, because that's what they're more familiar with, but the digital one and the nomenclature, they seem to find very, very confusing. If anyone has any useful feedback on why that might be, I'd love to hear it, because it's a real barrier. Yeah. Yes. Yes. Yeah. Yes. Yes.
And
yeah.
maybe just a graph.
Yeah. I mean, right. I mean, you can interpolate between these two, sure, but might help a little bit. Yeah. Okay. I could. Yeah. Yeah. Okay.
Maybe you could
version 2, but I'll try presenting and see if I get a better feedback. Anyway, yes, maybe that's one for beer or coffee afterwards. Anyway, okay. So what this talk's going to be about then is these two different modes, which exist in terms of gene regulation, and how they talk to each other. Okay, so the context is going to be this plant system, actually, that I've been thinking about for a long time now, because it's such a great example of epigenetics. And actually, in terms of what's understood in terms of individual epigenetically regulated loci, it's very well understood in comparison with lots of other loci about which we know far less. So what is this system? So it's a gene in Arabidopsis called flowering locus C. This is all work done with my long-standing collaborator, Caroline Dean. And what this gene does is, in terms of plant developmental biology, it's very interesting because it aligns plant development with the seasons. Because the most important decision that any flower ever makes, any plant ever makes, is when to flower, right? That's an absolutely critical decision. If this developmental switch is mistimed, so that it's done at the wrong moment in the seasons, the plant will flower and it'll still be snowing or something, and very poor probability of reproductive success. It's got to time this correctly to give a maximum chance of reproductive success. So there's the system that does this is integrated with this gene called FLC, which actually stops this transition. If FLC is highly expressed, then you don't get this switch. And there's a bunch of pathways that regulate the expression of this gene. I'm going to talk about two of them today. One is called vernalization, and one is which we now call transcription-coupled repression. So vernalization is the one I'll talk about first. And vernalization is the means by which this gene is switched off by prolonged periods of cold temperatures. So it's an environmentally mediated silencing system. If you give the system lots of cold, this gene will get repressed. And once this gene is repressed, it can't do its repressive activity. So you're repressing the repressor and you allow this switch to flowering, okay? So that's the idea. And this is triggered by cold. And that's the whole point here, really, that this gene needs to stay to repress this switch until the plant's had enough cold for the plant to decide, yep, I've had winter. I can turn off this repression and allow the plant to flower in the spring. Okay, right. So why do we study this epigenetic system? Well, first of all, why is it epigenetic? I mean, that's because you start off at this high expression level in the autumn. It then gets progressively lowered, the expression of FLC during the winter, and then it stays low in the spring. So what you can see here is that you've given the system, the plant, an environmental signal. In this case, it's cold. It's done something. It's turned this gene off. But the point is that once the signal's disappeared, the cold temperatures have gone, the repression remains. It doesn't, if this was just a normal system, right, you'd repress it and then it would go back up again once the signal had disappeared. But that's not what happens. The gene remains repressed even after the environmental signal has disappeared. So it's kind of got to be epigenetic because you've had a transient signal and then it's come back on afterwards. Is there a question? Put yeah. Yeah. Yep. Yes. Yep. Yep.
If I put it in an artificial environment and try cycling up and down the temperature in phase with that, what would happen?
Right. So I'll come back to that because we've done that experiment. We did that about 8 years ago, actually, precisely to try to get at what's it measuring? How does it respond when you start to kind of fluctuate the signal? Or is it still able to recognize it, or does it get reset by periodic periods of warm? So it's a very, very good suggestion, and it's what we did because and the way hopefully in a few slides' time it'll be clear how it's able to deal with that kind of you say it's a nasty experiment, but of course out there in the field that's exactly what happens. Right, you get a burst of warm temperatures in the middle of winter, and it's got to be able to deal with that. So yeah, okay. So here are these phases that we're going to talk about all of these phases actually if I have enough time. I might not get to the last bit talk in each case. That doesn't matter. Okay. So now I just want to say a little about epigenetic memory, about how do you hold memory of things in biology. So there are various ways of doing this. There's one form of memory that I haven't even got listed here, which you can refer to as like transacting memory. So it's basically circuits of transcription factors that auto-activate or auto-repress themselves. So that when those circuits get activated, because they have very strong feedback, they'll maintain a certain gene expression state. And the memory there is essentially held in the concentration of the transcription factors that mediate the network. And that's sometimes called a transacting network because the constituents that hold the memory are diffusible and they can move and they can act in trans. So not going to talk about that. I am going to talk about the sort of cis-based epigenetic memory, which come in two types. By cis, I mean they're sort of encoded in things that are associated with DNA. One is DNA methylation, and we've worked on that, but I'm not going to talk about that today. The one I am going to talk about, and which came up last week in some of the talks from Helmut and Leonid, are nucleosome modifications. So what are they? So nucleosomes, as I'm sure many of you know, are these proteins that package the DNA, and you wrap the DNA almost twice around each nucleosome particle. Each nucleosome consists of 2 copies of four of the core histones, H2A, H2B, H3, and H4. And these histones have tails that stick out to which you can add covalent modifications like methylation, phosphorylation, sumylation, acetylation. There's this enormous zoo of things you can stick on the tails of these histone proteins, and they affect gene expression. And in particular, we're going to be investigating this one, which is one that wasn't so heavily discussed last week. This is histone H3 lysine 27 trimethylation. And the system that adds those modifications is called the Polycomb system. So there's a whole suite of methyltransferases and other enzymes whose dedicated job it is that atoms subtract these histone modifications and thereby regulating the expression of the genes that lie underneath them in the sequence that's wrapped around these nucleosomes. And here's a kind of cartoon of that, some of these kind of barrel-shaped proteins, histone proteins, and out of them come these lollipops, which are the histone modifications. Okay, right. So now on to FLC. So FLC is an example of where these histone modifications play their role of holding memory of something. So the experiment here is a very simple one, is you have your plants and you put them in a cold chamber for a long time. So the black is when the plants haven't had any cold, and then you see orange, green, blue, and red as you give the plants more cold. So that is 2 weeks, 4 weeks, 6 weeks, and 8 weeks of cold, just stuck in this environment chamber at 5 degrees centigrade. And what you see is that over time, the levels of these histone modifications, in this case, H3K27 trimethyl, increases at the locus. So the more cold you give it, the more of these histone modifications you have. Yeah, yeah. What's the y-axis?
Yeah, the y and also axis.
Sorry, the x-axis is just the gene. So this is the FLC gene. That's the transcription start site. That's the termination. That's in base pairs. So it's about 6 kilobases long. Also interesting to note the scale, right? It's not very big. Maybe I'll come back to that at the end.
Sorry, the y-axis.
And the y-axis, this is just the scale in the experiments. This is called a chip qPCR experiment. So it's chromatin immunoprecipitation followed by, by amplification reactions. So it's not, I mean, you can also do this by, by ChIP-seq, and then you just zoom in onto the, but for many years in Caroline's lab, they didn't want to do that. They did this chip qPCR, which I think until very recently was probably more quantitative. Sarah. Yes, absolutely. Absolutely. So it flattens out after about 8 weeks. It reaches a saturating cold duration, after which you get no more. And essentially, I'll explain to you what's happened at that point. Okay, so that's very important. Actually, there's lots of interesting stuff here. I won't really be able to get into, like how long does it take to saturate? Well, it depends where your Arabidopsis plant is from, right? If you're an Arabidopsis plant from northern Sweden, it'll take more like 3 months to saturate. If you're one of the ones from further south, it takes less time. There's a lot of natural variation. It's adjusted how much cold you need to give the system before you get a saturating level, which is kind of interesting. Okay, so one immediate question, going back to what I said at the beginning, is this an analog or a digital response, right? So it looks like you've got this lovely, smoothly rising level of histone modifications. But that's an illusion, because the experiment that does this, you're grinding up like thousands and thousands of cells and measuring it all, and it's not a single cell experiment, right? So you haven't actually learned anything about what's going on in individual cells by doing this. So what's going on in individual cells is something we really need to understand. Yeah. Okay, so first a bit more about how these histone modifications get put there. I mentioned this is something called the Polycomb system, that comes from its historical origins in fly work. In the mutants of the system had extra features, and that's why it got called polycombs, because it comes from Drosophila. Anyway, the apparatus that puts these histone modifications here, it's called PRC2, Polycomb Repressive Complex 2. It's actually an amazing machine which is able to recognize existing K27 methylation, and then once that's happened, the whole complex becomes enzyme and allosterically activated, such that the active subunit EZH2 is then able to add more of this histone modification nearby. So, and actually has a quite a large fold up regulation when it's able to detect this. So what you can see is there's kind of a feedback element here. Once you have some of these histone modification done, it can add more of it nearby. Right. Okay, so I've argued at the moment that the memory of this system is stored in these histone modifications, H3K27 trimethylation, which, as we've seen at FLC, the more cold you give it, the more histone modifications you have. But of course, there's this problem with histone-based memory that I think was also discussed last week, which is that in some ways, histones and nucleosomes are an absolutely appalling substrate for memory, because every time you go through DNA replication, you don't have enough of them to fully populate the two daughter strands. So here's the parental nucleosomes, here's your replication fork, here are the two daughter strands. Oh dear, there's only enough to populate half of what you need. And worse than that, it's believed that the system that distributes the nucleosomes is essentially random, such that it just hands them out randomly to one of the two daughter strands. And that means there are going to be large fluctuations, because as I said a moment ago, the targets that have the memory are not very long, right? This is a 6 kilobase gene. It's got somewhere between 20 and 30 nucleosomes. So they're going to be enormous low copy number fluctuations every time it goes through DNA replication. Yeah. The PRC2, these ones. So their role is to find the histone modification and add more nearby. Their role is not to hold the memory. They're just the elements that actually add more of the modification in the appropriate places.
But can you not say that because their location will be close to that point, that would promote more histones to sort of, depending on where the spatial location is, that's where they will.
But it's believed that most of these proteins are wiped off at the appropriate stages of the cell cycle. But you're right, actually. I wasn't going to go into this, but we have other experiments that I'm not going to talk about, which indicate that the histone modifications on their own are not enough to hold the memory in these systems, right? We've known that for a while now. We have some mutants where you localize the histone modification to such a small region across the gene that only has a handful of nucleosomes in it. You'd expect the memory to be completely blown apart in a few cell cycles, and it's not. And that's deeply perplexing if you think the memory is only being held by histone modifications. Because if you, in this mutant we have, where we think there are only 3 nucleosomes, you'd expect a 1 in 8 chance of a complete loss, right? If it's random, 50-50 each way, 1 over 2 cubed, you've lost the whole lot, right? And all these fancy systems that actually recognize histone modifications and add more, well, that's no use if you've lost all the histone modifications because there's nothing for them to work on anymore. So we think there's something else going on, but I won't have time to say more about that. Yeah.
How long? It's the lifetime of the Polycomb repressor, normal cell cycle time.
So how long is the K27 trimethyl? Yeah. So it's believed that the K27 trimethylation is a very stable modification, and yeah, it doesn't get turned over very quickly. So as I said, there are all these different histone modifications. Activating histone modifications tend to have much faster timescales. So the ones that are associated with transcription, like K4 methylation, K36 acetylation, K27 acetylation, they have very rapid timescales of turnover, and they're actually believed to not be inherited at DNA replication. Right? Well, there's still some controversy over this, but there's some evidence to suggest that only the repressive modifications are inherited. Okay, there's this problem with memory. You lose half of it all the time, right? How do you deal with this? So we heard a little bit about this again last week, but I want to offer a slightly different perspective on it. Because one of the things we didn't hear at all about last week was transcription, right? And transcription is the whole thing that's being controlled by this system, right? The Polycomb system is trying to repress transcription. That's its function. And there's a lot of evidence to suggest that the act of transcription itself is antagonistic to these histone modifications. So here's a kind of cartoon for what we think of is one of the important things, not the only thing, but one of the important things going on at these memory systems. So you have this off state with the red lollipop, that's like the K27 trimethylation, and you've got these read-write feedbacks that are trying to make more of itself over here. And these red lollipops inhibit transcription. That's their function, is to stop transcription. But you've got transcription itself, and we think that transcription antagonizes this state, and it does that in a variety of ways. It can pop out nucleosomes, it can demethylate nucleosomes. There's a variety of different ways. It can titrate away the PLC2 components if you transcribe too much. There's a multiplicity of ways in which transcription really disrupts these silenced memory states. So the way we think of it at these targets is there's kind of a tug of war going on between an active transcription state and this Polycomb silent state, and they're really struggling with each other to establish supremacy. So if you take this reasoning further, then what the conclusion you come to with a bit of modeling that I'll show you a bit later is these systems are actually bistable. They're digital. You either have an on state or an off state, right? And that's all you can have here. It's very difficult to generate an intermediate state with an intermediate level of histone modifications because it's not stable through DNA replication, right? If you have a 50% level of histone modifications and you attempt to inherit, and that 50% means something, that means that when you go through DNA replication, on average, it will go down to 25%, but with big fluctuations. And then somehow the system has to magically know how many inherited and go back up to 50%. So multiplied by 2, but only on average. And that's an incredibly difficult thing for it to do. It have to basically measure how many nucleosomes it was inheriting and then do a computation on the spur of the moment while the replication fork is going past to shove the right number of nucleosomes back. Okay, so instead we think it's this digital system, and that means that if I go back to the FLC system and that system where you saw these system modifications were building up with time in the cold, what we're suggesting is that the more cold you give it, the more the greater the fraction of silence loci. So it's a digital system, and you just increase the fraction of silence loci the more cold you give it. So long ago now, we of course wanted to measure this experimentally, and so we did this through fluorescent reporters. And this is an Arabidopsis root, and what you're seeing here is that it's actually different roots, but after different periods of cold, 2, 4, 6, 8, 10 weeks. 2 weeks, everything's on, because remember, FLC expression starts high. 10 weeks, it's completely repressed by the cold. Everything's black. The interesting thing is what happens in the middle. You know, and what you see here is some cells that are completely black, some cells that are still yellow, right? As if they've been kind of completely switched off in some cases. And you may be wondering about this, kind of, you know, surely if this was, you know, an on-off thing, I should see some kind of salt and pepper pattern of completely scattered cells, some which are on, some which are off. Instead, you see these enormous files of cells, which looks, you know, really weird. Yeah, they look like they might be on or off, but where do the files come from? And to understand that, you have to understand just a tiny bit about the development, the growth of plant roots. And the reason why they get huge files is because the stem cell, it's growing downwards, and the daughter cells from these stem cells are aligning themselves in these columns above them. And the nice thing about plant cells, they don't move. Right? You don't have a problem with tracking them, which of course is a massive problem in other systems. So what happens is that a stem cell down here, which has or has not flipped its epigenetic memory state, all the cells directly above it just inherit that state. And so you see an immediate readout in the experiments, oops, here, of the epigenetic state of the stem cells here. So the reason why you get this huge file is there's a stem cell sitting there, which hasn't switched off. And right next to it, there's a stem cell which has switched off, and it's pumped out cells that are off. No, I often get that. I really should use some different pictures on this because we have other ones where it doesn't look, it's just coincidence, actually. I know exactly what you mean, but yeah, I mean, sometimes people, they switched off on one half, right? It's just a bit random because there aren't very many stem cells down here. There's only like, 10 or 12 cells, actually. And therefore, you tend to get these patterns. And sometimes they go in and out of the plane as well. Like this one here, you see some of it. That's actually because it's gone out of the plane of the imaging. Okay, right, done that. Right, so what's the kind of model that we have that underpins all of this? So this is our kind of foundational model that we published a few years ago now. And it consists of a sequence of transitions from a lowly methylated to a highly methylated. The highly methylated being the silenced one, and the lowly methylated one being the activated one. And we have these feedbacks that allow the highly methylated states to make more of themselves. We allow the highly methylated states to repress transcription, but then we allow the act of transcription itself to actually push the system in the opposite direction by demethylating, by popping out nucleosomes. So, and then we allow, of course, transcription itself to be regulated by standard transcription factors. So you can control how much transcription the system is trying to put through this gene by controlling the concentration of these trans regulators. Okay, so that's kind of our basic model of this. And you can, of course, add extra things. There's any number of extra things you can add into this. You can put in activating modifications. You can put in volume changes. You can put in transcription titrating away stuff. There's a lot of stuff that could, in principle, be contributing to this. But what I want to emphasize, I think, is that transcription is at the heart of a lot of this, because the system has evolved to turn off transcription. And we know that transcription antagonizes a lot of this. So I think it makes a lot of sense to build transcription into the basic model. So so we have reason to think that this is reasonable. Demethylators co-purify with RNA polymerase. And this is the sort of, very straightforward, Gillespie algorithm type simulation. It's hard to do analytics on these systems because there's a kind of mesoscopic number of components in it, which means the fluctuations are enormous, which makes it hard to do calculations. Yeah. So that's a very good question, and that's really badly understood. No one really knows what is methylation doing. I think the best guess is it's somehow just stopping elements of pole 2 from engaging and starting the act of transcription. But there's various points at which it could do it, right? It could, for example, inhibit the pause to the elongating state, right? It could stop the loading, or some point in between those two. I don't think anyone really knows that at the moment, how that works. So maybe someone in the audience knows more than I do about that. But it's a very hard question, it seems, to pin down. Okay, so here we're just going to make some assumptions that that's how it works, right? And see whether whether we get interesting results out of it. Okay, so here's a sort of simple description. This is just an algorithm for putting it into a Gillespie simulation where you can control fully for the stochastic effects. It's it's actually pretty simple, and this is the sort of output that you get. So you take the thing through the cell cycle. Here you've got this crash in methylation, which is DNA replication when you lose half the modifications, and it builds the modifications back up, right? Because it's got this huge amount of feedback in it, and therefore it can recreate this digital system, and therefore it's very robust to these perturbations. Okay, so one thing I'd like to emphasize of what you get when you combine this sort of regulation. So I've got kind of this read-write feedback in the system. But if I just go back to the previous slide, I've also got these transcription factors that can feed in that are trying to push the system the other way. So I've got a kind of digital regulation, but I can also regulate how much transcription factor activity I have in the system, because the transregulators feed into this transcription. And what pops out of that, if you do this, so this is a simulation of the system, is you get something which we refer to as the sys memory window, which sounds a bit too much jargony. But all it means is that there's a range of parameters over which the system exhibits bistability, right? So if you balance these two feedbacks, right, you've got this transcriptional feedback that's trying to make it highly expressed. You've got this Polycomb feedback that's trying to silence you, right? If those two things are roughly balanced, you're going to be able to make these alternate gene expression states, either quite silenced or quite expressed. But if you ramp up the transcription too hard, you're just going to overwhelm the system, because you're going to try and put so much transcription through it that you're going to break whatever memory system you've tried to impose there, and you'll push the system to a high expression state. On the other hand, if you have no gene activity whatsoever, it's just going to silence itself, and nothing will ever happen, because it'll just be permanently silent, because there's nothing to remove the repression. And that means there's an intermediate range where you get this memory window. And so what we're saying here is that this memory is not an absolute property of any given locus, right? It depends on how hard you try to drive transcription through it and a bunch of other factors. And I think this subtlety has not always been recognized in the field. It's not just you've got a target that always has memory. Well, it will have memory sometimes, and there'll be other times when it doesn't have memory, because you don't have the appropriate parameters at that target to endow that particular locus with memory at that time. Okay, so yeah, I said that. So now I'm going to take a slight aside. I do time, and leave the plant system for a moment. And we wanted to test some of these ideas and try to understand, well, is this true that transcription is really antagonistic to these spin modifications, and is it really going to disrupt the memory, a high level of transcription? So we switched to actually a mammalian system at this point. So this was a collaboration with Raphael Margeron's group at Curie, actually, in Paris. And the experiment that Raphael did, I think inspired by this model, was to look at what happens so this is now human embryonic stem cells and neural precursor cells, and look what happens when you took out the Polycomb system and then put it back in a sort of two-step process, okay? So often this happened, which is pretty uninteresting. So here you are before, then you do this experiment where you have inhibited the Polycomb. So you've inhibited the repressor, And unsurprisingly, loads of genes come on because you've taken away their repression. No surprise. And now you've washed out the repressor. And in these 213 targets, it's completely uninteresting because it's gone back to the state it had before you did the perturbation. So there's no memory in this system, right? You've perturbed it and you've removed the perturbation and it's gone back to where it was at the beginning. Okay? No, that means you don't have it because you haven't remembered the perturbation. What you want to remember is the perturbation. You don't want to default back to the initial state. Otherwise, that's not memory because you've only got one stable state. You have to have two stable states to have memory. Yeah, no, you have to think about it slightly. Yeah. Okay, but there's a bunch of targets that behave differently, right? So they start out not being expressed. You take away the repression, expression goes up. Now you put back the repression, but these targets stay high. So these ones have remembered the perturbation. So they have memory because they flipped their gene expression state as a consequence of the perturbation. So they're clearly stable in two states because they were stable in this state to begin with. You've done this perturbation, and now they're stable in this state to the end. Yeah, and this is quite a long wait. It's many days, so they're really pretty stable in this new expression state. So you've flipped them. So that's kind of interesting. It's also interesting that this property is not present in embryonic stem cells, which is only present in more differentiated cells like neural precursor cells or mass embryonic fibroblasts. And that possibly, when you think about it, is not a massive surprise because you might expect the embryonic stem cells to be completely naive and therefore not want to lock themselves into particular gene expression states. That's a property you would only want in more differentiated cells. So perhaps it's not a huge surprise that embryonic stem cells don't have this property. I don't think anyone understands why it is that embryonic stem cells are unable to do this, but the more differentiated cells are. I think that's a massively important question, which I'd love to know the answer to. Okay, right. So the obvious thing is what distinguishes these targets. There were this whole bunch here that were reversible, and these ones are irreversible. What's the difference between these two? Now, the model would suggest the one thing that's different is that perhaps these ones transcribe more, because the reason they're able to maintain this high expression state is because they have an innately higher level of transcription, so that when you put the repression back in the system, the highly transcribed state can still resist that repression. Okay. So interestingly, when we measured this, the one thing we consistently saw is that the genes that were irreversible, they had higher levels of transcription. And when you looked in more detail, they had things like higher levels of nascent RNA, and they had higher levels of histone modifications like K36 trimethylation that are associated with higher levels of transcription. So it seems to be true for the model prediction, this is from the model, that the irreversible ones should have significantly higher levels of transcription, actually seems to be true in the data. So that's quite encouraging for this idea that there really is something in this idea of transcriptional antagonism to the Polycomb silencing. Okay, right. Yes. So if you inhibited transcription as well, we didn't do that. Perhaps we should have done that experiment. That would that ought to. So I think the moment it matters is when you're going back. Well, what it's doing while it's so you'd have to do that quite carefully, right? So you'd have to have a transient burst of transcriptional repression as you're removing the inhibitor. That would be a pretty good experiment, actually. Yeah.
How many generations do you work with?
So it's about once a day, and the reason why it's 14 days is because when you inhibit PLC2, right, the stuff is just so stable, it stays there. So the only way to get rid of it is to dilute it out to DNA replication. So you've got to go through lots of cell cycles to get rid of it. Okay, maybe 14 is overkill, right? Because you should halve it each go. So, you're going to be pretty good after, 7 or so, right? Yeah. Okay. Right. Okay, so I think I've gone through all of that. Right, okay, summary. Right. So hopefully what I've convinced you is that Polycomb epigenetic memory is digital. So going back to the start of the talk, analog versus digital, epigenetic memory is digital. I only know of one case where it isn't digital, which is plant DNA methylation. And someone can ask me about that at the end, why that's not digital. I can go into that. And I've emphasized these read-write feedbacks are antagonized by transcription. Okay. So now I want to go on in the last part of the talk. So I've got another 15 minutes or so, is that? Yeah, something like that. Okay, is to talk about this other pathway, this transcription-coupled repression. So so far, I've only told you about this cold pathway, the vernalization one, right? More cold, more digitally silenced cells. So now I want to talk about this other pathway and basically ask the same question, right? Is this pathway analog or digital or what? Okay. And this is a complicated pathway. I have to see how far we get into the regulation of this. It's not like this other pathway is just regulated by some nice transcription factor. If only it were that simple. You could just, the system was dialing up or down, changing the concentration. Unfortunately, as we'll see in a moment, it's a bit more complicated than that, unfortunately. Nevertheless, it's mediated by an RNA binding protein called FCA, right? So FCA is very important in this other pathway, and we want to understand whether it's digital or analog. And the way we did this is we use what's called an allelic series. So there's a bunch of mutants of this FCA protein that are sort of intermediate in their effect, right? They're not complete loss of function. So we have one which is complete loss of function. That's this called FCA1 allele, and that's actually a premature stop codon, and there's no functional protein, and it's total knockout. This FCA3 case, which is actually a twice-like mutation. And if you look at the amount of FCA RNA that you make, you can see it's intermediate between the wild type, which is this LER case, which is very well silenced, and this complete loss of function, okay? So we've managed to manufacture an intermediate case, which has like half the level of normal expression. And the reason we want to do that is because we want to get at this analog digital question, right? Is it all or nothing or smoothly varying? And you can't answer that question if everything's already repressed or everything's active, because you'll just see lots and lots of cells with nothing in them or lots in them. And that's no use. You've got to have something that's in the middle so that you can either say, well, half of them are completely off, or they're all on, but only at 50%, right? So anyway, fortunately, we've got this through this FCA3 allele. So now we can ask the same question. So, right, onto more experiments. So we did this imaging in the roots that I showed you before. And in the previous case, it was in the cold. Now forget about cold. These plants have never seen any cold. This is all just in the warm. And we're just looking at, how much FLC is there in these roots. So this is the wild type, the lands, it's called LER. And it's supposed to be very highly repressed. And as you can see here, you see absolutely nothing in those cells, right? They're all completely off. This is the complete loss of function, FCA1. Very nice. You see, as advertised, the FLC protein has come on in all the cells, so it's completely on. Okay, so the question is, what happens in FCA3, which is this intermediate one? So it's a bit difficult to see here, right? Because you might know, is that one on, some off, right? It's a bit difficult to tell. So we enhance the images, which of course you have to be careful with. Well, you're enhancing images, you're not enhancing noise or whatever. But you can see that when you enhance the wild type, it started with nothing and you ended up with nothing. Right? If you amplify 0, you still end up with roughly 0. But if you amplify the FCA3, it's quite interesting, right? You definitely see some cells that clearly have some expression in them, that have some protein in them, right? You also see some cells that essentially have nothing in them, right? No visible FLC protein at all. So some cells still appear to be completely off, but others seem to express at an intermediate level, right? Which you could regard as analog, because they're clearly not as on as these guys, right? Just look at which were all collected under the same conditions, right? These are the fully on ones. These ones are nowhere near as bright. So it seems that we've managed to mix this analog and digital regulation in this FCA3 case. And you can quantitate that in more detail because you can put all these cells through an image analysis pipeline, and you can pull out and measure quantitatively their protein levels. But what you seem to get is this mixture of analog and digital regulation. In this case, some of the cells are completely off, but not all of them. But the cells that are still on, they've been dialed down in intensity, right, from their fully on state. So as you can see, it's a mixture of both. Yeah. Haha, yeah.
I mean, if you took this now later, would the cell the same protein?
Okay. So an hour later, yes. So we've tried to do time lapse, right? The problem is, and why I don't normally talk about that, is because it's not very reliable, because this is a lowly expressed gene under the best of circumstances. So if you try and acquire the images, you've got to turn the laser up. And what happens when you turn the laser up to get enough signal is that the cells become extremely unhappy and they stop growing, and they don't quite die, but they just don't do anything anymore. And we've got a couple of cases where, for reasons we don't understand, the root didn't do this and went on growing. And they do interesting things that I'll tell you about in a moment, okay? But on a time scale of an hour or so, you basically won't see any change. This is a slow process, but I'll come back to the dynamics just in a slide or so, actually. In fact, the next slide. That's very, very good question. So I think that's sort of what you're asking, is this really a dynamic process? And we were thinking, well, I know in the cold that this is a very dynamic process of switching off and when the cells go completely off. So are we also seeing a dynamic process here in the warm? And it took a ludicrous amount of time to actually realize this and do anything about it. But eventually we realized, well, surely we should look at this over time now, right? And so we did. And of course, when you just do this through measuring expression, of course, you start to see everything's going down over time. This is just measuring RNA expression. When you look in the roots and you do the imaging of the protein, you see the same. But over time, this is, I think, 7, 15, and 21 days. And this is the quantitation of the amount of protein, it goes down. And you can do the same with single molecule fish, and you get the same answer, right, if you measure the RNA levels. So everything's going down over time, okay? So this is sort of interesting. So what we're finding here in this intermediate FCA3 case is an interesting mixture of analog and digital regulation. That some of the cells seem to be completely off. The ones that aren't off have been dialed down in an analog way. But over time, those dialed down cells are all switching themselves off over time, right? Eventually, 21 days are very left on. So you've got a kind of dynamic switching. It's not regulated by culture. It's just over time, developmentally, these analog dim cells are switching off.
Is that the basis of adaptability?
So in the cold case, it would be. This is a slightly artificial system because it's been constructed by mutation, right? Doesn't exist naturally, because we wanted to understand how this pathway works, and I wanted to understand this fundamental question about analog versus digital. So we created this mutant. But there are other natural pathways that probably do the same things different way. Okay. Right. Okay, so how long have I got? Right, okay. So the last part of the talk is about the mechanism of how you make this analog reduction. And as I alluded to a little bit earlier on, I'd love it if this analog reduction was just some transcription factor that bound at a lower concentration and triggered a reduced level of transcription across the gene in the way that transcription do, and everything would be lovely and simple. And but unfortunately, that's not how it works. For whatever reason, this gene has chosen to regulate its level of transcription in this pathway, a much more complicated co-transcriptional mechanism that involves antisense and all sorts of other players. I don't want to get detail. I'm not sure that's going to be the greatest. It's a complicated pathway. Short termination, chromatin modifiers. So why the system has chosen to do this, I do not know. But what it boils down to is again some feedback loops between termination of transcripts, and the termination of those transcripts changes the chromatin environment through a different histone modification called A4 monomethylation. And basically, if you terminate quickly as you're trying to transcribe a gene, that recruits a factor that removes these activating modifications. If you remove the activating modifications, it encourages more early termination. So there's a feedback loop. If you start terminating early, it will do more early terminating, and you get a feedback loop. But it's not a particularly strong feedback loop. And it turns out if you build a model of this and it works on both strands, sense and antisense, which I mean know that because we've done something called Quantitative, which measures where you've got polypeptide transcripts. But it seems that you have this complicated model that is controlling the level of transcription in the analog state. But we can build a model of that right on the slide. The interesting thing is that this whole module is hooked up to this digital silencing system that I spent the whole of the talk telling you about. So here's a model that's just trying to regulate transcription, and it's coupled, because transcription is coupled to Polycomb silencing, this entire module is coupled to the digital silencing network. So what you've done is the system has made an analog module that can dial up or down the transcription of this gene. Right? You've seen that in the experimental data, and this module will do that. When you build a model, a biophysical model of that, and you simulate it, it does what I just said. But it's the digital silencing module that's trying to completely turn off the expression of this gene. It's complicated, and obviously the only way you can figure out what this sort of system does is if you take a very quantitative approach. So what you discover when we simulate all this is that these two modules, what they generate is a slow switch into this silent state. Right? You've got this module that's creating an analog level of transcription. This is digital, and the amount of transcription that you have from this module is controlling how rapidly you make this digital switch. Okay, so I think actually I'm going to go to a summary slide that's more comprehensible. So what we've got is we've got these different levels of analog transcription. Okay, and this is modulated by this transcription with the protein FCA. And this is your analog regulation, allowing you to dial it up or down depending on how good the FCA protein is at doing this. Then you've got this digitally silent state, which is the Polycomb silent state, where everyone's just expression gets crushed to zero by Polycomb. And now you've got switches between these two. And the key transcription antagonizes this digital state. If you don't have much transcription, you've got no antagonism, which means you switch very, very quickly because there's nothing to stop it. If you've got a higher level of transcription, you've got more transcription. There's more resistance to switching into this silent state, so it takes longer. If it's a very low transcription state, it completely prevents this switching, so that it never will does so only with very low probability. And that's really sort of the punchline here. That's how these analog and digital systems are combined. You've got an analog system that can control the level of transcription in an analog way. You've got your digital off state, and it's how much analog expression you have that controls the time scale for the switching. Right? If you have very little transcription, you switch fast. You have a lot of transcription, you don't switch at all. And if you put it in the middle, you can see the switch over a time scale that you can see experimentally. So this seems to be the way in which digital is combined in this system. And we think that this might be quite generally applicable, right? If you've got a cell-phate system that wants to make a decision about what direction it wants to differentiate in, what cell type it wants to become, you can control the time scale on which that developmental switch is made by altering the amount of transcription in the projected before it has a chance to differentiate and to actually acquire a particular cell fate. Okay, so that's the punchline. Transcription coupled repression controls the rate of Polycomb silencing. And that's how we think analog and digital regulation is combined in this system. Okay, so I think that's enough for the time date. So I just want to end by thanking the people who did this work. So what I've told you about is the result of many years of work. You can tell it's a long time because the people who were originally involved in this project now actually have their own groups. You can see that that's been a while. So this project was started a long time ago when Scott Berry was a PhD student in my group. He now has his own group in University of New South Wales in Sydney. Another student, Robert Eichwald, actually a senior postdoc in the Churchman group in Harvard. Rhea did a lot of the work on the more recent analog digital and the alleles work. She now has her own group in Glasgow. A lot of imaging was done by a PhD student, Sven Urik, who's just finished. And some of the more recent analysis, the model of the analog digital system was done by Govind Menon, who's still a postdoc in my group. There was a great collaboration with Raphael Margeron's group in Paris and his postdocs, Daniel Hollock and Michel Vasser. I'd just like to thank Caroline for the many years of fantastic collaboration, and particularly thank Eduardo, who did a lot of the actual data I didn't really show you at the end because I think it was all getting a bit much. And Stephanie Rosa in Uppsala in Sweden also did a lot of the imaging. And these organizations coughed up the money. So thanks very much for listening. So I think there's at least three ways. So first of all, transcription, the polymerase machinery just tends to knock out nucleosomes completely, because it has to be also an elaborate chaperone system to put them back in behind the polymerase. And that won't work with perfect fidelity. So it's going to lose some nucleosomes just because of that. Then we know, at least in the FLC system, because we can do the biochemistry, we can pull down the demethylase with POL2. So we can pull down a protein called ELF6, which is a K27 demethylase. The third way is, it's, I think, pretty well established that Polycomb repressor complex 2 binds promiscuously to RNA. So the more transcription you put through a gene, also you've got a transcript coming out back, it's basically going to hoover up PRC2 locally and take it away. So there's at least three ways in which you can do it. Actually, we only did the model. We did the model, so we didn't put it in. So I think it's plausible. But none of this explains how you put the K27 in the right place to begin with, which I think is really a pressing and really deep problem that no one knows. Right. So the spreading, that's an interesting one, because as you know, a lot of these systems work through nucleation and spreading. I've seen more stuff I haven't had time to tell you about. And FLC is exactly the same. You saw that in the K27 profiles, where they have this peak in particular. And they, the spreading process is very interesting. So we have two mutants that stop that. We can knock out one of them, it's called CLF, and then it won't spread. Or you can also knock out LHP1, which is, a version of heterochromatin protein 1. That also takes out the spreading process. So it can nucleate it, but it can't spread it. And it's very interesting that LHP1 has that property, because, that's involved in the state and so on. Interestingly, if you make that mutant, it won't do it. And moreover, this is probably more for the experts, but if you knock out what's called this chroma shadow, the one that binds to the effect, what it cares about is the RNA binding domain. If you knock out the RNA binding domain, complete loss of function in this context. Whereas it doesn't care about histone modifications. It only cares about binding RNA and probably therefore making some, confined droplet in that region. It's very interesting. I don't completely understand it. But I think there's a lot of scope for some interesting models. Yeah. Right. So it's interesting that. So yes, of course you'd think that the temperature would have a really significant effect on it. But obviously plants evolved to deal with that somehow. And actually it doesn't, so just putting something in cold temperature messes up their expression. They, plants have to deal with it on a daily basis, or most nights in the winter. So it doesn't seem that it's as simple as that, just affect transcription. And it is a very interesting role, how does low temperature to trigger this whole pathway? So we think we do strangely, but the protein's very long-lived. It doesn't get diluted, so its concentration goes up. So basically what the plant's done is it's coupled the temperature dependency of growth through a long-lived protein, creating. As soon as it gets into the warm, the plant starts growing, protein gets, so it goes away. So it's found kind of for free temperature sensor. So this protein has apparently no temperatures. It's not like, it has a different structure at low temperatures or anything like this. Its only thermosensing property is the fact that it lives a long time. And then it creates this indirect. Why it's very difficult to escape this, where everything's what's the feature which is doing it. So it's but there are other effects, and then nuclease another protein which comes down, which tips down at the nucleation site for where the K27 gets pushed into the genome. But there's so many things we don't understand, right? You can ectopically upregulate these genes in the warm, and it doesn't do it. Right, so there's an additional need for cold over and above the need to just have these proteins expressed in the cold. Right, it needs cold on top of that, and we don't know what it is. There's something else about the cold that's making it. Yes, now you can start this process, and it's a bit mysterious. There's so many things that are still mysterious about it, even after all this work. Yeah.
This is a general question with time scale with memory.
Yeah.
So, and we think that the transcription actually is set the time scale in the sense that only thing that is not transcribed.
Yeah.
And
Okay. No. Okay. Yes.
Cannot remember.
Yes.
From one winter.
Right.
To another. Right?
Yeah.
And someone will transcribe the gene. Right. So is the memory wiped out?
So the memory is wiped out. So Arabidopsis is an annual plant, so it's dead. Yeah, some variants are not annuals, and they live longer. So what has to happen is its memory has to be wiped out. And actually, even in Arabidopsis, which is an annual, it makes seeds, right? And the seeds have to have the memory wiped. Otherwise, the progeny will kind of germinate already thinking that they've had the cold. So the whole process has to be reset in the early embryo. So one of the things we've been looking at that I didn't talk about is when does this whole process get reset, right? And it appears to be very early in embryonic development. All of these marks are wiped away, and it starts the whole process again, right? And it seems to occur very soon after germination, right, within a day or two. The whole thing's wiped, probably by upregulation of demethylase that just take the whole thing away and blast it away. But your other question of time scales, and is it just it's not just the absence of transcription regulating this. You've got to be a target for these. Right. So and something is designating certain genetic loci as being targets for Polycomb. And that's not very well understood about what makes a target. We know that it comes in through nucleation and spreading, which we've described biophysically, but we don't really know what is it that actually makes the nucleation point for this modification to come in, because they're the ones that are going to be sensitive to to transcriptional dynamics and whether transcription will itself nucleation processes which start, and that's just not very well understood how that works. Yeah.
But earlier on you mentioned that the saturation point for
yes
for control is variable, and that's presumably that's genetic variable.
Yes, absolutely. So the way that do it is it varies the starting level, so that it can reduce the starting level to increase the dynamic range, and that starting level can be varied by non-coding genetic polymorphisms, SNPs, basically a small number of SNPs at the locus, nucleotide polymorphisms, which control apparently the starting level of this thing. And I think from an evolutionary perspective, that's a very tempting way of doing it, because, you know, it's just changing, you know, a very small number of local elements.
Well,
It-
that was looking for Lamarckian, so opening for Lamarckian parity, right?
Yes,
So for
Yeah.
I don't know, for a sequoia. that
Yeah.
there was a year, you wonder if it would actually be competitive
right,
to so slowly vary,
right.
you know, the level of saturation of
Yeah, okay.
hundreds of years.
Yeah, I mean, I doubt whether Sequoia has this machinery in it, but you know, it is definitely, there's some really interesting questions about how evolution ties into this and how you control the dynamic range. Actually, there's a question that Budo asked, I remember earlier on now, about the temperature variations that I didn't answer. I'll answer it for everyone else. Right? But the question that he asked was, what happens? And you can't really mess around with the temperature profile. But you see, the whole digital system just copes with that. Because what it's done is in the period when it's cold, it's digitally flipped some cells. Then when it becomes warm again, they've already flipped. It's too late for them to be affected by the warm temperatures. Then when it goes back into the cold, it just starts flipping a few more ones digitally. And over time, it just flips more and more, and it ignores the short-term temperature variations to high temperatures. So it's completely resistant to those fluctuations. And it has to be like that. Otherwise, every time there's a warm snap in winter, which we get more and more of, the whole system would be bust because it would erase all the memory. But because it's digital, no problem. It just remembers as it goes, basically. Okay.
Maybe we.
Okay. Yeah, we are. Yeah, of course we are. You know, I'm part of the program. I'll let you. Philip can let you in as well. You know. Yeah.